Welcome back. Today we're thinking about lithography, which is a way of patterning silicon wafers typically, but also used for other things. So if we take a wafer, if we take a wafer like this, what we want to do is to be able to pattern things onto it, to be able to make some kind of structures. But typically if we sputter, for example, we're going to get a coating over the whole thing. It's not very directed. If we etch, we also need to have some way of defining which bits are going to get etched and which bits aren't. And typically we would do that using a process related to film. So we're going to get some kind of sensitive paint on the top that we're then going to pattern. If we start off with a silicon wafer, we will typically have silicon hydroxide or silicon oxide at the surface, and that will have an influence on what we can coat it with. So as we had before, we would typically be able to coat this with polar things and with silanes, but in this case we don't want to coat it permanently, so we want it to be able to come off again, and we want to avoid an issue with water at the interface. We want to have a coating of typically very hydrophobic polymer, a very hydrophobic coating, and that is not going to interact very well with this. There will tend to be a water layer in between, and that will cause us trouble. To avoid this, one of the things that we can do is react it with a silane to make it into a trimethyl silyl group, and for practical reasons, the one that's used in this case is hexamethyl disilazane, which I'll draw now. So there it is with its two trimethyl silyl groups on it and the nitrogen, and this is substituted for one of the surface groups. This makes the surface then hydrophobic, and we can continue our process. It isn't always necessary, and in practice you can often get away without it, but if you need really, really fine detail, then it's often difficult to do otherwise. So now we get to our resist, which we have to coat in a very, very smooth manner. One of the ways to do that would be spraying it or to roll it on in a thin coating, but very often at wafer level it's done by spin coating. In this technique we have our surface and we rotate it very fast, a few hundred to a few thousand RPM, and we have our drop of resist, which is a paint, and it's spread out by centrifugal force, centripetal acceleration, and it causes it to thin down to a certain thickness. I believe the contents of that are in another course, so we will not cover that now. What's important, however, is that the photoresist has a solvent that evaporates at a reasonably moderate rate, because if it evaporates too fast, then spin coating won't work. It'll evaporate too quickly, and so it won't spread properly. If it evaporates too slowly, then we have a problem that it's sticky and we'll get all kinds of dirt on it as we're trying to dry it. So now the next step is we need to push our picture, so whatever we want to have, right up against it, and then shine a bright light on it. So the plan now is to put something on there that has places that let through light and places that don't. Shine our light on it and expose, convert the photoresist or paint into another form in these areas. So for that we need to do some chemistry. There are several main types of chemistry. We'll have a quick look at 2.5 of them. The group that's going to absorb the light is the same one in these two, the nitrogen that is got a partial charge imbalance, so got a plus here and a minus here. This one's got 3 nitrogens and this one's only got 2. We'll start with this one because it's actually the slightly more common type. The nitrogen is going to try to leave when we excite it with light. So we're going to excite an electron out of the double bond into there to make a triple bond, and that will cause a rearrangement. It causes a rearrangement called a Wolff rearrangement, which moves this carbon up to behind this oxygen. Let's go through the reaction in a few steps. First of all, this double bond is going to go into here and do the standard thing that carbonyl groups do, which is absorb an electron pair temporarily. Let's resolve that. That gives us this species, which is effectively nitrogen attached to our molecule, and so it's obviously going to be very unstable. Our electron can kick back in, kicking something away, and we're going to lose this onto the nitrogen. That seems reasonably likely, but instead of moving these bonds, we have a shift of this carbon across. So before bending it back into a better position, that will be where our carbon was, and our bond has flipped from here to the next one, giving us an extra carbon in our chain and a 5-membered ring instead of a 6-membered ring, and this strange double bonding situation. So this is our product of photolysis, and now we're going to make it react further. And the typical way to do that is with a weak base in water. So we're going to try to dissolve our photoresist with a weak base in water, and that will react here to form a carboxylic acid. So here it is being attacked by an OH minus, knocking out an electron onto the oxygen and then back. Let's that step, but that probably happens, and then it breaks this bond to form a new species. This obviously immediately, or at the same time, takes a proton, something else, to form the carboxylic acid. Carboxylic acids are charged, particularly in base, so it will lose a proton in the basic solution, and then it will be relatively soluble in water. So now we can make our insoluble organic compound, which was just hanging around the surface because it's not polar, into a more soluble one. This is mixed in with our polymer to make a fairly soluble polymer. The other alternative is to go the opposite direction. So if we take this species with 3 nitrogens and we do the same thing, so we shine a light on it, cause nitrogen to be released, that will leave us with a new species, which is a nitrogen with only 1 bond. This nitrogen is obviously gas, and so it's a very strong driving force for the reaction, and this nitrogen on its own is extremely reactive then, because it's really unhappy only having 1 bond, and it goes along and attacks a neighbouring molecule. Typically that will cause our polymer to cross-link, so we have these on the end of some polymer, and it will typically cause it to cross-link. This agent is also used in biology to make things go hard. So if we have a protein sample, or we have a body or something, and we want to make it stiff, then this is one way of doing it, that we can react it with a reactive nitrogen molecule that then cross-links the polymers, so DNA, collagen, proteins in the body, making go hard. It's often used in biology for preparation. So that was the 1.5. Now we've got the extra one. We have a strange antimony salt, an antimony fluoride, that's connected to these strange ligands, the sulfur benzene, so there's benzene sulfide, and we excite it with some light, and that generates this new species, which is hexafluoroantomonic acid. As we can see, so going back to hard and soft, and the acidity of things, as you can see, the hexafluoroantimony ion is extremely stable because it's extremely large. The negative charge on the fluorine is very stable, and the antimony is happy in the middle, and that makes it a very weak bond to this proton here, which has the effect that the hexafluoroantomonic acid is unbelievably strong. It's called a superacid because the hydrogen is really not connected at all. So we generate it in a photoreaction, and that makes a proton. It makes a very, very strong acid, and that can catalyze reactions. In the particular photoresist that we're going to use for this, we have an epoxy group. The epoxy group, as we know, is very unstable, but it can't just react. And now what we're doing is we are adding an H+, which can polarize this bond, allowing it to be opened very much more easily. So then that makes this epoxy even more reactive than it normally is, and then we can selectively react the ones that are protonated. Because we get the proton back afterwards, it's more a catalyst than an actual reagent, because our other cross-linking agent and the other agent that breaks or that solubilizes it is only reacting once per photon. In this case, we've got an enzyme. We've got a catalyst, and that makes the reaction different, because it means that if we react it for longer, we can have more effect. So we've got another control parameter. That's good and bad. It means that we can vary our process and get different results, but it also means it's hard to get a good process. If we have another one of these, or a hydroxide, so an alcohol-ending polymer, it can react with this when we warm it up. And if we warm it up, we can allow the protons to move around, and we can allow the chains to move around a little bit to find each other. That will cross-link it and form the equivalent of a very large polymer. Then we can use the normal solvent, the solvent we put it on the surface with, we can use in exactly the same way to remove it again, because the other part of our material is not affected. So we have the two possibilities. We have, let's take these pieces as the pieces we've exposed, bits that are here, and they're either cross-linked with the tri-nitrogen cross-linking agent, or we made them more soluble with the 2-nitrogen agent, or we've used SU8, which is also going to be hard at these points eventually, but we have another step to do in that case. These are normally called positive and negative photoresists, and it's terribly confusing which one is which. The positive photoresist is the one where this becomes soluble. So first of all, nothing happens inside our light source. We take it away and put it in a bath of sodium hydroxide, which removes these pieces. In our negative photoresist, we do exactly the same thing. We take it away and we put it in the bath. The other things go away. So we get the opposite of our pattern. This is obviously important because we need to plan what we're going to get afterwards. The reason that this is all important is that usually our next step is to do something, is to either etch, in which case we're going to affect these uncoated regions, or to coat something else. In which case we're also going to affect these uncoated regions, but we will affect the top as well. Let us do, in green because I don't have another pen colour, let us do a coating all over using a sputter, and that will mostly coat the top here, coat them both, and the bottom here. Should have done that in red, shouldn't I? And now, if we get it right, we can remove this and be left with the piece we want. Obviously, because sputtering is done at a medium pressure, we need the pressure of the argon typically to sputter our metal or whatever we're shooting. Typically, then it can go round corners, and there is a danger that we'll coat the sides. So, if we want a photoresist to do this, we will usually organize it so that it hangs over a little bit. Should draw that the other way around. Draw it the other way around here. If it hangs over a little bit, because it's better at shading it, then there will be a bit left for the solvent to attack. We can remove these pieces left with the bottom. This is a process that requires quite a lot of optimization when you're setting it up. Because obviously it matters whether you have the top the size you want or the bottom the size you want, and what you actually get is somewhere a little bit in between depending on what your code does. So now I've removed it. I've removed photoresist using that second process because the positive photoresist is still soluble. That's one of its major advantages. The negative photoresist are somewhat more difficult to remove because we've cross-linked them. So we are going to have to do something more exotic to remove them. The epoxy-based ones are very, very difficult to remove, and so usually you leave them in. The other ones you can get out, but typically you have to use quite harsh reagents. Here we're going to have a patterned surface of some kind of coating, either chemicals that we want to use later, semiconductor maybe, or conductors. The important part for us, however, is more the chemistry of the process, because you might be ending up formulating these. And also the side issues, of course, like if we use our SU8, our cross-linking epoxy, it's often used in biology, but one should always remember that there is an antimony ion still dissolved in the polymer that might make its way out, and so that is going to be a potential issue later.